Let's recap what you've just learned. The previous two exercises have given you a glimpse of the later stages of a typical ChIP-seq workflow. In this video, we'll talk a bit more about the results you can expect to see from a ChIP-seq analysis and how this will help us to better understand the mechanisms driving the observed differences between our two groups of samples. One of the first questions you want to ask of a dataset like this is whether there is evidence for a systematic difference between groups. This heat map of sample correlations clearly shows that samples form blocks according to the group they belong to. Notice that group membership is indicated by red and blue bars along the side of the plot. Plots like this one are useful in assessing sample quality. You'll learn more about how to do this in the next chapter. Knowing that the correlations between samples conform to the expected patterns is useful, but doesn't really tell you much about what the differences between groups are. Looking at the height of individual peaks across samples can be a bit more informative. This heat map shows different samples as columns and different peaks as rows. The color of each cell corresponds to the height of that peak. As you can see, each group has its own set of high and low intensity peaks. You'll learn more about how to compare peaks between groups in Chapter 3. To gain a better understanding of what the observed differences in protein binding actually mean, it is very helpful to associate observed peaks with genes. This plot visualizes overlap in genes associated with peak codes in the two groups of samples. Each of the vertical bars corresponds to the size of one subset. The dot below the bars indicates which groups these genes were observed in. As you can see, many of the genes that are associated with peaks in one condition don't have any peaks in the other. This provides you a list of genes for each condition that can serve as a starting point to investigate the real differences between primary and treatment-resistant tumors at a molecular level in much more detail by uncovering common themes among the functions of these genes. You will learn more about how to do that in Chapter 4. Now it's time to take a closer look at how to do all this.